The overall goal of this procedure is to purify intact, highly active yeast ribosomes. This is accomplished by first lysing the cells and clarifying the lysate by high-speed centrifugation to obtain the S30 fraction. A cysteine-charged sulfo-link column is then used to purify the ribosomes. This is followed by removal of contaminating tRNAs with puromycin treatment. As a final purification step, the ribosomes are subject to high-speed ultracentrifugation.
The main advantage of this technique over existing methods, like ultracentrifugation purification, is that use of the sulfo-link column rapidly separates ribosomes from contaminating exonucleases and proteases. Further, the puromycin treatment step separates ribosomes from contaminating RNA species such as tRNAs. These steps ensure production of large yields of highly active, intact, purified ribosomes for use in biochemical and structural studies. Demonstrating the procedure will be Jonathan Leshin, a postdoctoral research associate from my laboratory.
Begin this protocol by pipetting a 50% slurry of sulfo-link coupling gel, as supplied by the manufacturer, into two 10 ml plastic vials, with 5 ml distributed into each vial. After briefly centrifuging the vials at 850 times G, carefully remove the supernatants. Wash the gel 3 times in coupling buffer by resuspending the beads in 5 ml, and then centrifuging briefly at 850 times G. Remove the supernatant by pipette. After washing the resin, add 5 ml of a 50 millimolar solution of L-cysteine in coupling buffer to each tube.